Okay, so look at this respiratory substrate. Okay, most of the time we say that we use glucose. Okay, it's an essential respiratory substrate for the cells. Basically means that we use the glucose most of the time. Okay, that's why we need to have the carbohydrate. Okay, now, but the thing here is, can we use other kinds of the substance to become the respiratory substrate? The answer is yes. We can use lipids. We can use the amino acid. Okay, so how about lipids? Lipids, basically we hydrolyze the lipid into glycerol and the fatty acid. Glycerol, we know that is a 3-carbon, so it means that we can convert the glycerol to the glyceroldehyde 3-phosphate. So it means that glycerol actually we can actually convert them into the triose phosphates, okay, that we learned. Then it will continue from there. Okay, how about fatty acid? Fatty acid, we know that they have a lot of the carbon, right, and hydrogen. Hydrocarbon, correct or not? So they do have a lot of carbon hydrogen bonds here. So what we go do, we're going to oxidize it so that it form acetyl group. Can you see that? So it means that this acetyl group can enter into the link reactions. Right? This acetyl group will combine with the coenzyme A. So this oxidation is termed as beta oxidations. Beta oxidation on this fatty acid actually break down, okay, this fatty acid into the 2 carbon fragments, which is called acetyl group or acetate, which can enter into the carbon this Krebs cycle as acetyl coenzyme A. Okay, and we do know that fatty acid contain higher, okay, proportions, okay, of hydrogen atom. So therefore, lipids, when you look at the lipid of fatty acid, they give us a higher energy density. Okay, later we're going to look at this. Okay, so amino acid also present in excess. Okay, so those amino acid that we eat too much of them, we're going to have, okay, we can convert them, okay, to intermediate of glycolysis and Krebs cycle. But before that, we're going to do deamination. So what's mean deamination? Deamination, basically we remove the amino groups. Okay, so we know that this is the, okay, HR group and COOH and H2. So deaminations help us to remove these amine groups or these amino groups. So the remaining one, we can convert them into the intermediates. Okay, either intermediate in the glycolysis or Krebs cycle. So this is how we actually oxidize them. So first we have to carry out deaminations. After that, the remaining, okay, the remaining one is called keto acid. So these keto acids can be converted. It depends, okay, what are the R group and everything. So they can be the intermediate of the glycolysis. They can be intermediates of the Krebs cycle. Okay. So this diagram summarize the various respiratory substrates. So we do know that you can see that a carbohydrate. So this carbohydrate, if let's say is a glucose, they enter the glycolysis. If let's say it's a fructose, then you can halfway through. Okay, from here we can enter here. Okay, from fructose become fructose 1,6-bisphosphate. Okay, then after that, from here we get the glyceroldehyde 3-phosphate. Glyceroldehyde 3-phosphate actually is a thiophosphate. So from triphosphate, get the pyruvate. So we have done for the glycolysis. And then we get the acetyl-CoA in the link reactions. And then you have the Krebs cycle, oxidative phosphorylation. So this is a normal way. But how about the fat? So the fat actually is triglycerides. So for triglycerides, hydrolysis takes place. We are going to get the fatty acid plus glycerol. So this glycerol, because 3-carbon, glycerol 3-carbon. So this 3-carbon actually can be converted into a triose phosphate. Can you see that? We can convert them into triose phosphate, and from triose phosphate, we complete the oxidation process and then enter the link reaction to form acetyl-CoA and then Krebs cycle and then oxidative phosphorylation. Okay. How about fatty acid? Fatty acids are quite long hydrocarbons, so we break it down in this beta oxidation process, make them into the 2 carbon fragments, and then attach them with the coenzyme A. So then what happens? Then they can enter into the Krebs cycle. Amino acids, a protein, we break down, hydrolyze the proteins, we get amino acid, we carry out deaminations first, okay? So the remaining keto acid depend, it can be converted to pyruvate or converted into the intermediates. Some of them, they can be converted to acetyl-CoA as well. But most of them, actually these two, okay, either converted to the intermediates of the glycolysis or intermediates of the Krebs cycle. Okay, so what about the energy value of the respiratory substrates? Who give us the higher energy value? Okay, so this energy value is termed as the energy density. So it means that per unit mass, okay, means that we have the 1 kg of oil and 1 kg of the carbohydrates, which give us more energy value or energy density. The answer is lipids. Okay, so you compare, lipid have give us the highest. Okay, so most of the energy liberated in aerobic respiration come from oxidations of the hydrogen atom. Okay, remember, we remove the hydrogen atom, oxidize this hydrogen atom to water. Okay, when the reduced energy and reduced energy are passed to the electron transport change. Are you clear? Or we reduce the water. Okay, reduce the O2 to water. Okay, we oxidize hydrogen atom or we reduce the oxygen into water. Okay, so hence the greater the number of hydrogen atom in the structure of the substrate, it gives us higher energy value. Okay, so fatty acid, we know that have more hydrogen per mass compared to the carbohydrates do. So therefore, lipids will have higher energy value per unit mass, okay? Or we call it as a energy density. So if we look at this, if I have 1 gram of lipids compared to 1 gram of carbohydrate, then you realize that the energy release here will be higher compared to the energy release by 1 gram of carbohydrates. So how we know about this? Okay, we can always use this, what we call as the calorie meter. Now be careful, C-A-L-O, calorie. We measure the calorie, not calorie. Calorie meter is to determine the color intensity. So this one is a calorie meter. So what we're going to do here is, now, so we place our sample here, okay, in this chamber. Okay, this chamber will have the oxygen supply. Then we have the ignition vial to actually burn our sample. Can I see that? And because when you burn, the heat energy release. Are you clear? So this heat energy will be absorbed by the water, and we're going to see the increase in the temperature. So the energy release can be calculated. Okay, so energy release can be calculated by MC theta. Okay, M stands for the mass that we put it in, mass of the sample. C is mean the specific heat capacity. Of water. Okay, so this is a constant. Okay, this one is a constant. Okay, and then theta is the increase or change in temperature. So by doing this, you'll be able, okay, let's say I put 1 gram of the, okay, 1 grams of my lipids, and I put 1 gram of my carbohydrates. We repeat the experiment. Okay, so the stirrer here can help us. A stirrer can help us to make the water temperature here evens. Okay, and then you can see that the temperature actually increase. Okay, so we have the insulator so that the heat won't lose too much to the surrounding. Okay, so in these aerobic respirations, then we'll be able to measure how much heat energy release per grams. Okay, so this table, you have to remember at least 2 of them. Okay, lipids versus carbohydrates. carbohydrates. So lipids, 39.4 kilojoule of the energy released per gram of lipids compared to the carbohydrates. So question always asks why. So first, why lipids have higher energy density compared to carbohydrates? So first, we talk about this. They have more hydrogen atom. They are lipids. They have more hydrogen atoms per unit mass. Or you can say that they have more CH bond per unit mass. Are you clear? More hydrogen atom or more CH bond per unit mass. So in this case, so during oxidations, more reduced NAD can be formed. So it means that during oxidative phosphorylation, so because we have higher reduced NAD, so it means that more hydrogen atom can be released for ETC, correct or not? So it means that we can generate steep protons gradient. When we can generate the steep proton gradients already, then basically means that we increase the rate of ATP synthesis. Are you clear? So this is why lipids actually has higher energy density. But the thing here is because you won't be happy if I ask you to take 500 grams of carbohydrates, you may be okay. But I ask you to drink 500 grams of the lipids, you will feel like very disgusting, and then you want to vomit out, right? Okay, because we are not used to actually take in lipid. Okay, so with this, I've done for our class for this respiratory substrate.